Hi to everyone. We are going to be presenting the presentation comparing photosynthesis, cellular respiration, and fermentation process, and we are from group 306. Hi to everyone. My name is Alicia Sanchez, and I will be talking about photosynthesis. Photosynthesis is the formation of chemical energy stored in the glucose bonds and oxygen releasing in the presence of sunlight. During this process, light energy is captured and it converts water, carbon dioxide, or minerals into oxygen. So we can clearly see in the model that light, carbon dioxide, water, and carbohydrates are being in the plant. We can clearly see the arrows, and oxygen is being released of the plant. There are two stages of photosynthesis, the light-dependent reactions and the independent light reactions. For the light-dependent reactions, we have that sunlight is transformed into chemical energy thanks to two molecules, the ATP and the NADPH. These are used for the synthesis of high-energy storage molecules, which is used in the dark phase. We also have that they are organized into photosystems 1 and 2, where one system collects the light and the other carries the electrons. The range of absorption of sunlight is wide and depends on the pigment that the plant has. Next, we have the independent light reactions, that they use NADPH and ATP molecules synthesize and dissolve in the stroma that surrounds the tricaloids. It provides the energy for the synthesis of glucose from CO2 and water. Next, we have the 4 stages of the Calvin cycle. The first stage is the carbon fissection, in which 6 molecules of it combine with CO2 form 6 atoms that react with water and give 12 molecules of 3 atoms of PGA. Next, we have the G3P synthesis, in which PGA is transformed into G3P. Next, we have the RUBP regeneration, in which 10 of the 12 GSP molecules regenerate 6 of RUBP, and the other 2 will be used for this glucose synthesis. Finally, we have the C4, in which it binds CO2 and 4 carbon compounds that increases CO2 to the Calvin cycle. The plants that do this are known as C4 plants. Finally, for photosynthesis, we can see the keywords that are the stroma, which is the internal cavity of the chloroplast, the ATP, which is the adenosine triphosphate, which is an essential chemical compound since it is used by cells to store and release energy. We have the RUBP, which is the ribulose bisphosphate, and PGA, which is the phosphoglyceric acid. And we can see an image of the Calvin cycle with its 4 phases.
Hello there, my name is Maria Yamila Quiroga Caldera, and I'm going to explain cellular respiration. Firstly, cellular respiration will be defined or is how we derive energy from the food that we eat, specifically from glucose. It is known to be a mirror image of photosynthesis, and it happens in the mitochondria. Okay, so we have cellular respiration, and it is divided into 3 cycles or steps. We have glycolysis, Krebs cycle, and electron transport chain. In the right image, we have the steps, or what it's called the outline, on how it is formed with the enzymes and everything. Okay, continue with cellular respiration, and here we have some key elements or facts that should be taken into account. And it's that glucose is catabolism, and it performs or leads us to glycolysis. Then it takes place in the cellular cytosol. Then glycolysis generates 4 ATPs, and in addition to that, it results into 2 pyruvates and 2 energy cells of NADH. A pyruvate will be defined as an enzyme that generates acetyl, and that occurs into the Krebs cycle. Finally, that'll lead us to electron transport chain, and that results in the synthesis of ATP, which gives us a net worth of 38 ATPs. Finally, we have some diagrams that synthesize what cellular respiration is and explain more detailedly what I have been saying in the past 3 slides, which will be the process of cellular respiration consisting on 3 basic steps. The first one is glycolysis, which is known for glucose being catabolism and performing its job, which takes place in the cellular cytosol. Then we have the step 2, which will be the Krebs cycle, and it basically just produces or generates a cytosol CoA. And lastly, we have what will be called the electron transport chain, and it's the synthesis of ATP with a net worth of 38 ATPs. And that'll be all.
Hi to everyone, my name is Daniela Elizabeth Siguel Martinez, and today I'm going to talk about fermentation. So, now we're going to talk about fermentation. Fermentation is a process in which molecules of glucose are broken down in an anaerobically form. Louis Pasteur used the term fermentation as a way to describe the changes in molecules and yeast that grow in the absence of oxygen or oxidation. Fermentation has a product of ethyl alcohol and carbon dioxide when fermentation is produced. The glucolysis in fermentation. Well, glucolysis in fermentation, it talks about the breakdown of glucose or sugar molecules. And when this happens, glucose is catabolized, as you can see in the equation. And as a product, we have that the outcome of iolysis will be lactate. As you can see in the images of lactic fermentation process, the sugar molecules are broken down to the ATP molecules to produce lactic, and this will help to the muscle. Lactic fermentation, we use it in our body, and our body produces it to reduce muscular and then produce glucose once again. Through the years, men all over the world are discovered from discoveries that helped to do artificial things. Now, in these days, we can do fermentation, which is called industrial fermentation. This is the one that is produced by men, and the industrial fermentation can create a lot of fermentation products depending on the sugar and glucose. As a result, industrial fermentation can have as a product ethyl alcohol, acetic acid, monoacetyl glutamate, cellular, and butyl alcohol. Now we have the keywords of fermentation. We have oxygen. It's a keyword for fermentation because it's important to know that fermentation does not need oxygen to be carried out. Then we have lactate, which is a product of the glucolysis that doesn't use oxygen. Lactic fermentation occurs in the lactate, and it leaves the muscle and returns to the liver, which is transformed in glucose again. Then we have a keyword that is fermentation, which is enzyme catalyzed pathway cells using an anabolic breakdown of glucose molecules. And then from another keyword, we have ATP reactions that actually are in the transformation of sugar in muscle, yeast, plants, and some bacteria. Thanks to the ADP molecules, the glucolysis can be carried out, and we can transform sugar into what I said, muscle, yeast, plants, and some bacteria.
Like reactions occur in photosynthesis as well as Calvin cycles. It occurs in the chloroplast, and the ATP is generated through photo cellular respiration occurs in the cytoplasm and the mitochondria. Fermentation uses anaerobic processes, that means that doesn't use oxygen. Changes in molecules of sugar break down, and lactic acid and ethyl are produced as a product of fermentation. These two both begin with glycolysis. During their process, ATP is produced for the cell to use.